Well, welcome to our next session. I am not Garrett Nash. He is in the building, but we can't find him. So I'm actually going to get the session started. And I think next time we should put tags on all of our speakers so we never lose them again. So this next session, moving on from what we talked about earlier this morning, which is like the current state of affairs and how we currently think about it. Although we did kind of move into some of the future visions of treating appendiceal cancer this morning. But this session is really going to focus on novel pathways, what the current roadblocks are in treatment and also in research, and also some of the lessons that we may be able to learn from other diseases. Nicole.
Okay. Hi, everybody. I'm Nicole Aguirre. I am here to talk about pre-targeted radioimmunotherapy, which is and our novel two-step method using a self-assembling, disassembling antibody, which I'm going to be calling PRIT and SADA throughout the rest of this talk. Okay. I have no relevant financial disclosures. Many people on my team do. I just want to very briefly talk about what PRIT is exactly. So PRIT is a pre-targeting method where an antibody is injected into the body, and it has one arm that can grab onto antigen and another that grabs onto a DOTA radiometal complex, so a radioactive isotope, which is injected at a different time point. The goal is to give tumoricidal doses of radiation to tumor with very little radioactivity given to background organs, such as, most importantly, kidney. So this bispecific antibody that we use is actually a well-developed method over decades. It was initially given as a one-step process, so an IgG with a radioisotope attached. The effects of this were catastrophic on the rest of the body, unfortunately. So people started to break it up into this pre-targeted or multi-step process, which decreased toxicity to the body, but often required a third step, a clearing agent step, in order to wash unbound antibody to decrease toxicity. We recently, in the past few years, have discovered an antibody that we think can do this in 2 steps alone. So SADA is that antibody. It has, just like every other antibody that's been used, one arm that grabs onto antigen target and one arm that binds the DOTA radiometal complex. But there's been an added 30 amino acid motif derived from p53 that gives this ability to assemble and disassemble. So when bound to antigen, it assembles into a tetramer, but when unbound in blood, it disassembles into dimers and monomers, which clear via renal filtration in a matter of days, so 48 hours, really. In order to apply this to appendiceal cancer, obviously, we have a lot of things that we need to do first. So what we really need to do, which has been discussed in the previous talks, is establish a tumor bank and some patient models. This is underway with collaboration with the labs of Dr. Karuna Ganesh and Dr. Eliza DiStancina. We are developing organoids and PDX models. We are also starting to stain these models, IHC staining, in order to see, do they even express the antigen targets that we're interested in? So namely, right now, what we're looking at is GPA33, which is expressed primarily in colorectal cancer, B7H3, HER2, and we do have a large repertoire of SADAs that are available to us, so we can expand this as time goes on. So after we have proven that we have antigen expression in our models, we're going to move on to a PRID, which is a diagnostic modality of immunodetection with imaging. We can do this with a large number of radioisotopes shown here using either PET or SPECT imaging, where in just 4 mice, we can see if our model really does target what we think it will. And then after we've confirmed that, we can move on to a therapy study with both beta and alpha therapies. So our beta therapy isotope of choice will be lutetium-177, and our alpha therapy is actinium-225. We do have a repertoire of appendiceal adenocarcinoma slides available to us, and we have stained these, a small number of them, for our antigen targets of choice, and we see that they do stain positively for all 3, GPA33, HER2, and B7H3, so these are all viable targets for appendiceal cancer. What I want to focus on today is a project I'm actually working on right now, targeting GPA33 in colorectal cancer. So GPA33 is a glycoprotein that's expressed in 95% of colorectal cancers. What makes it special is it's not endocytosed and it's not released into blood. So this makes it an ideal candidate for our pre-targeting method because our antibody can be held on the tumor surface while the unbound antibody washes from the body, and then we can inject our radioisotope and have it find that antigen. So for this colorectal project I'm working on, I used the radiohaptin actinium proteus. The reason I chose actinium is because as an alpha particle, it has a very short range of action. It's actually only 50 micrometers. So it has very little damage to surrounding organs. And it has a long decay half-life of 9.92 days. So it's a very long action in the body. Proteus, which is my kind of haptin holder for actinium, is shown here on the left. The yellow portion is the lutetium benzyl DOTA ring. That is recognized with picomolar affinity by the anti-DOTA arm of SADA. And then on its own in the body with no antibody present, actinium proteus washes out in less than 2 hours. So in the bottom left there, you see a graph that shows that at 1 hour post-injection, the percent of injected activity per gram left in a mouse's body is less than 1%. So to first start this study, I did a serial biodistribution where I injected a mouse and then studied it over a period of days. And really what you can see here is how high the activity in the tumor is compared to surrounding tissue. So using that calculated dosimetry, so for a dose of 37 kilobecquerels of actinium, which is a rather small dose, we gave 30.88 gray to tumor and only 2.56 gray to kidney, which is the organ of interest because it's how everything clears, so it is most likely to be damaged. The therapeutic index here is 12.1. Okay, so we first went with a very small treatment study. Just 74 kilobecquerels of actinium proteus was injected into mice. Controls either received no treatment or therapy alone with no antibody. At 140 days, these mice were looked at for histological changes and damages to surrounding organs. The kidneys showed minimal tubular regeneration and vasophilia in about 1 to 3% of the mice. Comparing this to a historical study where actinium IgG was used in a single step, greater than 50% of those mice had moderate to severe kidney damage. So it's a huge difference in toxicity profiling. So we decided to move forward and actually just dose escalate to see if we could cure some of these mice. So this experiment is currently underway. We gave 1 and 2 cycles of SADA-PRIT 1 week apart. And here we see what happened to these mice. So our control groups, we had 3 control groups, all passed away within 21 days. Tumor growth was quite rapid. SW1222 is a colorectal cell line that is known to be very aggressive. All of our treatment groups, even our smallest dose of 37 kilobecquerels, did regress, but our single cycle groups recurred rather quickly. And then our 2 cycle groups are still alive at this time, but are slowly recurring, except for our highest dose of 74 plus 74 kilobecquerels, which shows no evidence of disease at 105 days. So this is one of our measures of toxicity, which is weight. All our mice have steadily gained weight throughout the experiment as they age. On the left, we have a group of mice that are non-tumor, just toxicity controls to test how toxic this drug is with no tumor present. We did hematology weekly and then monthly after the first 4 weeks and have seen no hematological evidence of toxicity. So in conclusion, our 2 cycle groups correlate with improved tumor response. We see no clinical toxicities by weight or by appearance. We see no hematological evidence of myelo or renal toxicity. Pathology results are obviously pending because many of these mice are still alive. MTD has not yet been reached with this dose escalation. And that is it. This work wouldn't have been possible with a lot of help.
Thank you so much.
Let's go back. Or I'll start here. So good afternoon, everyone. My name is Kerry Benenato. I'm the CSO at 76 Bio. 76 Bio is a small biotech just over a year old focusing on developing a new platform for targeted protein degradation. And so here, as where I'd love to dig into a lot of science, it is very early days. So what I would like to do today is really just give you an overview of our concept and really why we're so excited about the strategy and where we think it could be very applicable. So I'm going to start with a little bit of a personal background. So about 8.5 years ago, I was introduced to mRNA as a therapy, as a concept. So I'm a chemist by training, background, organic chemistry. I started doing small molecule medchem. But after about 7 years, I joined this little company just up the street called Moderna. And really what I was hired to do there was, they were at the very early days outside. It was recognized that for this technology to actually come to fruition, we're going to have to solve a number of problems, and most specifically in the delivery, and that was really where I focused. And so go back 8 years, this was a pretty crazy concept. There's a lot of challenges that were going to need to be solved. One, obviously, RNA is immunogenic. If you can solve that, how do we deliver it? And then also the scalability and the repeatability of this concept as a drug product, probably one of the more complex drug products that I think we can consider. I don't need to belabor the point, but RNA as a therapy, I think, has been shown to have a very global impact with the production of the COVID-19 vaccines. This is a safe modality. It is effective modality. And the rapid to discovery development timelines, this is a repeatable process. It took from less than 3 months, 66 days from sequencing of the virus to phase 1 being dosed. Obviously, a very unique situation, but in terms of this as a potential drug product for a variety of diseases, there's a lot of potential there. And that's the thing where I had the opportunity to live that and be a part of that. Another part that I've been able to be very closely connected with is RNA as a potential to treat those patient populations that have no other options. And so here, a lot of work in the rare disease, and RNA is now at the point where it's having clinical success in a variety of different applications, either rare metabolic disorder, the work that Intellia is doing with CRISPR, and now being inhaled in a number of CFTR programs. And so that brings us to what are we doing at 76 Bio. And so here, what we are doing is we're using the power of RNA delivered in lipid nanoparticles to develop a new class of targeted protein degraders. So TPD as a modality, if you take maybe what the general concept is and what has been being pushed forward over the past several years in the small molecule concept, basically what you're doing is you have your target of interest, your molecule binds to your target of interest, and then is able to recruit the E3 ligase such that then it can drive the proteasomal degradation. Now, there's a variety of different challenges with this. One, just doing small molecule drug development, it's really hard to find small molecule binders to new targets. And on the other hand, for this to work, you need to recruit that ligase, the E3, such that you get the E2 and everything is scaffolded perfectly such that you can drive that ubiquitination process. And when it works, it's very powerful, but it's definitely really challenging. And I think that's what the industry is seeing. And then I think the other piece you have to it is potential of off-target toxicity. You're trying to develop orally bioavailable drugs, which are going to be able to see throughout the entire body and then potentially have off-target effects. And so what we're doing is we're using RNA to encode active fusion proteins, where here what we have is we have a target binding a domain of our molecule, which basically you can have antibody specificity, so we can use a peptide or a single domain antibody against a very specific target. And so here at the bottom, this is a computational model of one of our lead degraders bound to its target beta-catenin. And so in the red portion, that is the peptide binding domain, which we've been able to optimize and rationally design to increase the activity. And on the other side, that green part, that is an active ligase. And so what we have is we have our very selective target, and then we have an active ligase. This is an active E3. And what we're doing is we're not just appending wild type E3 onto different targeting domains. These are actually minimal pharmacophore that is needed to recruit the E2. And so what we've done at 76 Bio is we have built a proprietary library of these ligases and a very efficient screening platform that allows us to very rapidly identify leads to any target we're interested in going after. So what we're able to do is able to selectively target and degrade very historically challenging intracellular targets such as transcription factors. Another really uniqueness to our modality is that we can use localization sequences as part of these fusion proteins in order to localize our molecules, or our deltex is what we call them, to the specific subcellular compartment. And so on the left-hand side, these are some of our initial programs that are in a variety of different stages. And what we're able to do is selectively degrade nuclear beta-catenin. So with beta-catenin in its oncogenic activity is its presence in the nucleus. And it's not present in the nucleus in many other cells. And this is where the power of the delivery comes in that it is not going to widely distribute. So in terms of off-target toxicity and thinking about intestinal crypt cells, lipid nanoparticles are not going to go there. And so what some people think of as a bug for LNPs is actually a really unique aspect of selectivity for us. We were also able to enable the degradation of multiple targets. For example, we have a dual degrader that can degrade both beta-catenin and PCNA. And what is coming along very, very quickly and is not too far behind beta-catenin is we have a c-MYC program. And so where we are in a stage is we are currently in vivo efficacy models. We're really excited to push these programs forward over the course of the next year. But really, what is it going to take for this to truly be a broadly applicable technology? Our first indications will focus on HCC and colorectal liver metastases, because that's what the technology can do today. We can very confident we can efficiently deliver to liver tumors. But in terms of thinking about expanding our technology, that's where a lot of the innovation is going to have to be, and that's what we need to invest in and will invest in internally, and specifically on the delivery technology, optimizing these lipid nanoparticles to be able to go extrahepatic targets like lung, like heme. And this is not the most straightforward thing to do, but based on what I have seen in my experience, this is a very, very tractable problem. And so we're definitely really excited to expand the technology, because the broad, not just in the oncology setting, the inflammation and immuno-oncology, this is a platform that we're really confident can be really powerful. Thank you.
Okay. I was going to suggest on the fly that maybe we change the order, because what I'm going to say is very different than what has preceded, and I think what might follow. And maybe my comments are more logically tied to the first panel discussion. So I'm Keith Flaherty, a medical oncologist at Mass General, 23 years in the field, focused on therapeutic development in melanoma for my whole career, and first half of my career, kidney cancer, parenthetically. The relevance of the melanoma piece to this discussion is that in melanoma, which is not a rare cancer, cutaneous melanoma, we actually have rare subtypes of melanoma, and I would call out specifically uveal melanoma, or ocular melanoma, about 1% of all melanoma. That's rare, and tracks quite nicely with appendiceal carcinoma in terms of frequency. And so I'm just going to make this brief comment. We can expand on this in panel discussion as anyone likes to dig in further. It strikes me coming out of the first panel, given my experience in melanoma and particularly ocular melanoma, Lots of people were ignoring ocular melanoma. It was too rare to focus on, really, in terms of building an academic career. Companies weren't interested in targeting ocular melanoma specifically. It took us a while to figure out in the field that ocular melanoma marches to the beat of a different drummer than cutaneous melanoma. But around the year 2009, 2010, we figured that out. It has different somatic genetic underpinnings. Turns out that about 90% of ocular melanomas have an activating mutation in GNAQ and GNA11. So think of GNAS in appendiceal carcinoma as like a GTPase. That's actually highly related. So 90% of tumors have that. So we can use NGS to refine that, but the vast majority of patients have that. And then nearly everybody who has metastatic ocular melanoma has a BAP1 deletion / loss of function mutation. So we got this kind of aha moment in terms of somatic genetic insights. But basically, you look at a tumor suppressor like BAP1, you don't know exactly what to do with that. And GNAQ and GNA11 were just like GNAS in that when those were discovered, everyone said, well, they're undruggable, let's move on. Tried some indirect targeting approaches. Any case, here's why I'm bringing up the uveal melanoma experience, like dovetailing into appendiceal carcinoma. We needed to form a community. That was step one. Like until we formed a community that was focused on uveal melanoma, and it turned out to be a subsidiary, a subset of people focused on cutaneous melanoma at large centers in particular, where we had enough of a focus and interest across a set of centers that we began with patient advocates to actually form a community and like as a group pay attention to what was happening in the field. So that was step one. Trying to bring companies to the table is its own long discussion. Maybe we'll get into that a little bit. But step one is form a community. And now I'm going to make a few more points in terms of like steps 2, 3, and 4, at least as I think about like the rare disease problem more generally. So let me just pick on academic medical centers for a moment because I'm at a big one. What are we good at? And what are we really good at? We're good at going very deep in terms of understanding what's happening in patients with new therapies when those new therapies are being brought forward already because the private sector wants to bring those forward, whether it's pertinent to a rare tumor type or it's pertinent to a molecular subtype that spreads across cancer and might be represented in tumors like appendiceal carcinoma, things that are coming through the pipeline. We recruit those agents in, we try to be strategic, and then we throw the kitchen sink at trying to understand mechanisms of action, mechanisms of resistance, try to figure out who the patients are who best respond, the ones who don't respond and why, try to inform next generation therapies that could do better than the one in front of us, and ultimately rational combinations. And I've been saying that for 23 years and counting, and we have very few rational combinations to show for our efforts. But we have published more than 100 papers in Nature, Science, and Cell and New England Journal of Medicine describing the output of what I'm showing here. We're really good at that. Is throwing this approach at a academic medical center, or a handful of them even, at appendiceal carcinoma going to move the needle? Not alone. I mean, certainly not. That's not going to be the solution. It could be part of a solution eventually once you actually have a pipeline of agents coming in that are well matched to the disease. So KRAS targeted therapies, for example, in appendiceal, I would say, okay, fine. Those are going to come beyond G12C. The DV and other drugs, they're coming. And appendiceal carcinoma patients should get access to those therapies. We'll talk about that a little bit more. So one of the other things I've done is been a co-principal investigator of the NCI MATCH trial. And that started in about 2015. And we learned a lot in the course of that study. It wasn't a massive success in terms of curing cancer, but wow, did we create a platform. And once we did so, the demand was outrageous for enrolling patients in this platform. The fastest accruing clinical trial in NCI cooperative group history, flat out. I mean, 6,000 patients accrued in record time. But on a per month basis, it was astounding. Now, partly because we were investigating dozens of therapies simultaneously in phase 2 format. So single arm phase 2 trials are quite popular, easy to understand. Drugs have been to a degree de-risked in phase 1, and putting patients on phase 2, multiple phase 2 trials, pretty simple. So we launched this study across the network of centers, quote unquote, who plug into the NCI some way, somehow. But when I say centers in quotes, I mean, yes, there are big academic medical centers and smaller academic medical centers, but there are the so-called NCORP sites, the community oncology-based research program, NCORP. and community sites that aren't even at the scale of NCorp. And more than half of our accruals came from the community, NCorp and beyond NCorp sites. So we had massive demand from the broad community. So when we talk about decentralized clinical trials, this is kind of the first example of something that partially fits that definition. A long way to go. And we accrued a massive percentage of patients with rare tumors. So we were worried we might have to cap accrual to common cancers because we worried they would overrun the trial. No, it turns out that's not what happened. Why? Well, because there are clinical trials for common cancers and there aren't clinical trials for patients with rare tumors. So not surprising in retrospect that across big centers on out to community-based sites, we had a huge flux of patients coming to this study who had rare tumor types. So having a platform like this, could we sustain one like at the national scale, would be a fabulous partial solution to what any given rare tumor, including appendiceal, could benefit from. Unfortunately, though, this trial, accrued massively at a rapid pace and closed. And there's some successor trials, but they're really biting off relatively small subsequent questions. And the NCI has not committed to using taxpayer dollars to fund an ongoing platform for rare tumors, although we in the cooperative groups have suggested that would be the smart thing to do. In other words, let the common cancers do their thing. And for rare tumors, let's keep the platform open and roll more therapeutic hypotheses onto such a study. But unfortunately, that's not happening currently. If people in this room want to join me in advocating for that, I'd welcome it. Okay, so here's the final two sort of just quick little vignette data slides, and then I'll kind of draw my conclusions and save the rest, of course, for panel. So how do we jump from where we are currently in terms of having relatively few therapeutic insights? So KRAS inhibitors are coming, fine. You don't as yet have GNAS inhibitors, but I'm assuring you they will come. I've seen both GNAQ and GNA11 potent selective inhibitors preclinically emerge in the hands of companies. Unfortunately, they're pausing in terms of their proceeding because they're worried that the market is going to be too small, because each of those are about half of uveal melanoma, and that's about it. GNAS is going to probably have a similar fate. The chemistry part's going to be solved, and then the newly formed community that I'm suggesting is the starting point needs to figure out how to broker a deal with a company that is able to initially preclinically develop a GNAS inhibitor, get it in. Anyway, so beyond that, the dark matter that you see when you look at NGS data for appendiceal cancer, RNA sequencing, similarly, doesn't point you to any one, or large blocks of therapeutic strategies to investigate. So how do you jump beyond that? Functional diagnostics. So functional diagnostics have been the thing I've been preaching about for, let's say, 8 years now. And here's a version of one, ex vivo functional diagnostics. So you take a tumor biopsy or a resection specimen alive, and you immediately subject it to testing with drugs. You can use other discovery platforms that aren't per se drugs, but just drugs. Drugs that are available, meaning they're approved. They're post-phase one. By available post-phase one, I mean like drugs that have been somewhat de-risked that you might be able to get access to for a patient. In these types of platforms, you can investigate monotherapies, multiple of them, combinations, multiple of them. This platform that I'm showing you here actually includes the immune cells that come along in the biopsy specimen and is able to interrogate tumor immune interactions. So for anybody who wants to object, well, that this is only useful for tumor cell intrinsic vulnerabilities, no, doesn't appear to be the case. This general strategy, lots of different technologies emerging here. They are massively underinvested in the field. But I would say for trying to make big leaps forward from what NGS could give us to what comes next, I would suggest that in any tumor type, and definitely true for rare tumors, this is the type of strategy that allows one to nominate, like for individual patients, even therapeutic strategies, but certainly for appendiceal, like sort of systematically. Then there are fewer of these, but in vivo functional diagnostics. So really cool device that MIT investigators developed, and as you can see described here several years ago, that is being clinically investigated academically, where a micro device is implanted in a tumor about 2 days before a surgery is going to be done. Can that, in fact, even be extracted with an IR guided biopsy, so with a magnetic sort of beacon that allows one to locate it. In any case, this thing elutes drugs in a very controlled fashion. So you can put about 12 drugs into this little micro device. And what's beautiful about it is you get in vivo insights in terms of efficacy of therapies. It doesn't really matter what the modality of the mechanism is, small molecule, large molecule, tumor cell intrinsic, immunotherapy, microenvironment in between, just fine. It can only handle a certain number of therapies. And so I've made the case that we need both the ex vivo strategy and the in vivo strategy to actually really start to laser in. And here I'm talking about ways of actually understanding the potential therapeutic opportunities for an entire indication like Appendiceal. But this actually is work that is ultimately directed towards individual patient therapy selection. And for anybody who wants to object that that's just like, crazy moonshot comment, I would say, no, we actually do ex vivo functional diagnostic testing every day when we select antibiotics for patients who have bacterial infections. This is not that crazy. In fact, there are a few CLIA labs in the country that are already doing ex vivo functional diagnostics for cancer patients. So this is a strategy that at larger scale can absolutely make a difference. So I'm going to conclude my kind of opening comments by just making a couple points here. Continuing to do DNA sequencing, I think, is inarguable. That will allow the appendiceal cancer community to understand how KRAS inhibitors operate in this tumor type and ultimately try to understand the limitations of that and what rational combinations are needed that might be different than colorectal cancer. I'll grant that. GNAS mutations, as I've suggested before, you're going to keep finding those and those will become druggable. Okay, fine. That's what DNA sequencing seems to offer for now. RNA sequencing has been massively under-leveraged in the field in terms of nominating therapeutic strategies, but for both immunotherapy and antibody-drug conjugates, I would suggest, insist, like this is the era where that's happening. But for this to become part of the workflow, again, patients have to be molecularly characterized, and you have to be able to connect patients with treatments. Ex vivo functional diagnostics and in vivo functional diagnostics, I'm trying to suggest that if appendiceal cancer is sufficiently complicated where one drug approaches are never really going to make a big difference. I get it. I mean, I was at the Society for Neuro-Oncology asked to preach about like how to try to make leapfrogs in glioblastoma. Appendiceal cancer is not quote unquote worse than glioblastoma in terms of aggressiveness and lack of treatment options. And there I was like heavily hammering on some of these same points, but particularly the opportunity to leapfrog the one drug at a time investigation with functional diagnostics. Lastly, and this is where I could say a lot, but I'll say just relatively little, in rare tumors, you have to be able to learn from every patient. So create the community first and foremost, and then you need to have something that the term registry is not quite appropriate for. Registry is the beginning, but you need to have a way of tracking what patients receive and what their outcomes are. There might be exceptional responders to available therapies, lovely, and learn from those in terms of the molecular characteristics of those patients and their tumors. But for anything novel that's being tried, at the N of 1 level, and I would just strongly suggest that one not set horizons beyond the N of 10 level, right? Small cohorts, quote unquote cohorts, that are not a single center conducting a clinical trial, but that are a therapeutic strategy that's being tried in 10 patients wherever they might be across whatever jurisdiction. I was focusing on the US with NCI Match, but beyond the US if need be. You need to be able to capture that and learn from those outcomes. I mean, I'm preaching this emphatically because we don't do it well in any area, solid tumors, rare tumors. In uveal melanoma, we do now have this. We actually did create a registry. It's a funded thing, and now we're able to capture some of the insights that I'm alluding to for the first time ever. So I think that needs to happen and make much more fluid the concept of a clinical investigation. So clinical trial is like, conjures up certain notions of like engagement with the company and a sample size calculation. And you got to, if you don't treat 40 patients, you're never going to learn anything. I'd say for rare tumors, no, like don't hold your breath on that. I would say what you need is an environment, a community in which therapies can be explored, dabble with, based on the molecular characterization, ex vivo functional diagnostics, individual therapies offered to patients under single patient INDs. That's actually a very good use of single patient INDs to try new approaches, not looking for bunt singles for the baseball aficionados here. You're not looking for small effects in rare tumors. Like you just can't, the math doesn't work out. You have to be hitting for doubles, ideally home runs, which is to say if you treat 10 patients and 2 patients respond, That's worth further investigating. And then you do a proper clinical trial. But you gotta lower the activation energy, I would suggest, in rare tumors. And that's where I've been focusing a lot of my time and energy in figuring out how do we do that? How do we make more fluid access to therapies and break down this definition of what we think of in terms of clinical investigation? Okay, I'll stop there. Thanks.
Okay, thank you. Thank you for the invitation. It's really fascinating hearing about a new subject for me, and just having all the experts in one place is never a bad idea. So I am the Chief Scientific Officer at Aracari Biosciences, which is my conflict because I'm also a professor at UC Irvine. And what I want to tell you about is microphysiological systems. These are complex three-dimensional models of tissues that we are developing, and I think might have some applications here. So these are the well-known problems with preclinical testing. A lot of drug discovery over the years has been done using monolayers of monocultures. So taking tumor cells, growing them on a plastic dish, and it doesn't take much imagination to realize that that plastic dish in no way models the environment that a tumor cell usually finds itself in. The consequence of that is that gene expression changes. When gene expression changes, drug responses change as well. A colleague of mine in the industry told me this once: mice lie to us all the time. Turns out it's not that hard to cure cancer in mice. We do it all the time. It's much, much harder to translate that to the clinic. Reasons for that are many, but mouse physiology is very different to human. They're a fantastic model for a lot of things, but in some cases not so great. So this was a big moment for all of us working in microphysiological systems. This was the FDA Modernization Act 2021, and for the first time, organ chips and MPS platforms were called out specifically as something that could be used when filing INDs. So we hope that this is an area that's going to grow, especially with the 3Rs initiative, which is really big in Europe. It's growing here. For many, many reasons, people want to use fewer animals. To do that, we need better in vitro models. So this is what we do. We're trying to hit the sweet spot between the simple but not physiologic and the definition of physiologic, which is the person, but obviously not easy to use. We've mentioned issues with mice, monolayers. A lot of you are familiar with spheroids, organoids. These are great, way better than monolayer, but there are some issues with those as well, which I'll touch on. So what we've developed are what we call vascularized microorgans. And that's what you're seeing in the middle here. In red are living human blood vessels. In green happens to be a tumor. And so what we can create are perfused tissues. Dimensions of this are a couple of millimeters in either direction. So we're going to create an actual human tissue in a dish with perfused vasculature to support that. So you all know what this is. Your vascular system's a high pressure arterial side, a low pressure venular side, connected by capillaries. Capillaries where all the action is, drug delivery, nutrients, etc. We schematize it this way with a microfluidic artery and vein in red and blue. We have this diamond-shaped tissue chamber that connects them. We create a living vascular network in there, which anastomoses with the artery and the vein, and then we can build any tissue we like around that, including tumors. We've now put all of this on a 96-well plate format. There are no pumps, no valves, no computers to run it. It's all gravity-driven, so it's very simple, easy to set up. It's just a picture of the vasculature that forms. And the question is, well, why do we go to all of this effort to put blood vessels into these tissues? All organs are vascularized. Every cell in your body is within 100 microns or so of a blood vessel. In many tissues, the vessels actually help to pattern the tissue, so they control gene expression. All nutrients and drugs are delivered through the vasculature, so if you want to understand how drugs get into the tissue, having blood vessels makes sense. And immune cells are delivered to tissues and tumors through blood vessels. So if you're interested in CAR-Ts, gamma deltas, NKs, you need to know that they can actually get out of the vessels in the tumor and then kill the tumor. Just putting those cells onto the tumor in the dish gives you half the story. And the other half, can they actually get to the tumor, turns out to be critical. So, to best model human disease, we need vascularized and perfused tissues. So just some pretty pictures of it. Here you can see the tubes. Here we're running blood through. Yep. This is blood flow in the skin. You can see a very similar pattern. There's areas of high flow, areas of low flow. So in all ways that we can measure, the vascular system that we create matches what you would see in vivo. So we've used this in many different tissues, but in particular for this audience, we're doing it with tumors. So within these platforms, there's vasculature here in green. There are stromal cells, cancer-associated fibroblasts, etc. Normally, we don't label them, so you don't see them. So up here, you're just seeing the vasculature in red, the tumor in green, but there is matrix and stroma in there as well. Some of these stromal cells wrap around the vessels. They're pericytes, smooth muscle cells, and the vasculature will grow in and around the tumor as it does in the body. We've yet to find a tumor that won't grow in this platform. When we do grow them, they all have these unique phenotypes. Some are tight clusters, some are highly invasive, triple-negative breast cancer. Glioma is interesting. It really follows the vascular niche as it does in vivo. And recently with Dr. Senthal, who's here, we've been doing peritoneal carcinomatosis, and I'll talk about that in a moment. Key point here is that drug responses are different in 2D, 3D, and spheroids. So here's an example. The drug doesn't matter, we can talk about it later, but this is growth of a tumor in our platform, and all doses of this drug blocked tumor growth in the platform. If we put these exact same tumor cells in monolayer or spheroid, the drug has no effect. We know why. The drug happens to target the tumor stroma. The point being, if you're doing screens, you can miss really interesting drugs if you're just putting tumor cells in a plastic dish. And in many cases, if you use spheroids, you can miss the importance of the stroma. We've done lots of different drugs, lots of different tumors, published data. This happens to be colon cancer. Again, the growth of the tumor in black, in presence of the standard of care drug, we block tumor growth very nicely. We can reproduce this in a spheroid. But if we look at triple-negative breast cancer, again, this is a highly invasive tumor. It's invasive because these cells have lost adhesion molecules. Because of that, they won't form spheroids. So we can look at drug responses very nicely, but you try and make spheroids with these cells, they just fall apart. Again, because they're highly invasive tumors. How well does what we do mimic what goes on in vivo? This was a study we did and was published. In red is the growth of a colon tumor in a plastic dish. It grows rapidly, overgrows the dish very quickly. Put in drug, you can completely kill the tumor. The black line and the blue line is the normalized growth in a mouse and in our platform. They're overlapping. I was shocked about how good this was, so we did it again with a completely different tumor, got the same result. And again, with drug, you can see we control the growth, but we're not completely destroying that tumor as you do in a dish. When we look at gene expression, you can see gene expression in the tumor, in the VMT, a vascularized microtumor, matches very nicely with the tumor in a xenograft. But gene expression in the monolayer is completely different, which presumably explains why the drug response is so different. We can do single-cell seq out of this platform, and here in vivo, we get 3 populations of tumors, so we can see the heterogeneity, what we call A and B. There's a stem-like population. We get those exact same populations in our platform. So what can we do in the area of appendiceal cancer? So we have already developed a model for peritoneal carcinomatosis. This is something that Dr. Senthal came to me and asked, could we do it? And so we said, yeah, this sounds like a fun thing to do. So we redesigned our platform. So we have our vasculature here, and we have a peritoneal cavity here that we can address. We line it with peritoneal mesothelium to recreate the peritoneal surface. And then we can put in tumor cells, colo-205 here in orange. Those will adhere to the surface. And in some cases, they will migrate through that mesothelial layer where they can then interact with the vasculature. The key point of this is we can now test drug delivery either through the peritoneum, through the vasculature, or both, and see different tumors, different patient samples, which route is going to be optimal. So we're pretty excited about that. So what we'd like to do is use a similar kind of model to look at appendiceal cancer. So what are the requirements for us to do this? We need tissue. And I can't emphasize enough how hard it is for me as a basic scientist to do translational research without a fully committed surgeon. If you have one, it's so easy. If you don't, it's almost impossible. So working with Dr. Santel has just been a magical experience, I have to say. You need a good platform. We have that. I've shown you that. And a plan. So the plan is to do drug combination screens. And I recently, just waiting for the check, an R21, looking at combinations of drugs. So there's a paper published in Nature early last year from Matthew Garnett's lab here in Cambridge, saying, isn't this interesting? We've tested thousands of tumor lines, hundreds of drugs, and we saw very little synergy. What upset me about that was the entire study was done with tumor cells growing in plastic dishes in a monolayer. And as I said, I just don't think that's representative of what's going on in the body. So what we're going to do is reproduce that on a smaller scale in our platform. We picked colorectal, breast, and prostate. I'd love to do it with appendiceal as well. And then the idea would be test FDA-approved drugs, de-risk drugs, find what works, what combinations work, and then hopefully the physicians here could run with that. That would be the goal. All right, last minute. We are developing a personalized oncology platform. We take biopsies, put them in our platform, grow them up, test different drugs, get sequencing data, put it all together. Ultimately, we would like to give those recommendations back to the physician and say, this works in this patient, this doesn't. We've got 3 trials ongoing, one with Dr. Santel, so hopefully that will work out. So to summarize, tissue and tumor microenvironments are complex. We can't rely on 2D systems anymore. They just don't work very well for most drugs, most tumors. 3D systems are required. Mice are not people. They're great, but they do have their drawbacks. We believe vascularized tissues are the way to go. It allows you to look at vascular tissue interactions in the tumor microenvironment. We're doing stuff with patient tumors. Critically, vasculature allows you to deliver immune cells. We've looked at PBMCs, NKs, gamma deltas, CAR Ts in this platform. We can deliver them all. We can see killing. It's pretty cool to watch. We've got a bunch of other models too if you're interested. These are the cool people at the company. I don't have a slide of all the people who developed this technology at UCI, but my co-founders, Steve George and Abe Lee, have been in on this from the beginning, developing this platform. So thanks very much.
Well, good afternoon, everyone. And thank you, organizers, for giving me this opportunity to speak about clinical proteomics. So as a company, so I represent a company called mProbe. We are a CLIA certified, CAP accredited, and also New York certified lab that does clinical proteomics and will give the information to the oncologist for therapy. So that's pretty much the gist of this company. So just to orient everyone, because everyone's always heard about DNA and RNA, we are the guys that look at the protein side of things. So DNA and RNA work is done phenomenally well by a lot of commercial companies out there, Foundation Medicine, Tempus, you name it. Whereas our approach is that let's take a look at the protein. And proteins are one of the drug targets. That's almost all your drugs are towards a protein or at least influenced by a protein. So why not look at the protein? So in our goal was to go and play in the same space that the pathologists play in. We don't want to create any more hassles for anybody, just get into that same sandbox. So just a little bit complex slide, but this is the clinical proteomics workflow that was developed 15 years ago, patented technology and all that stuff. Once an oncologist puts an order in, we work with the pathologist team to get the tissue block in-house. More often than not, especially if that order is from an university system, yeah, good luck. Then we have to send the kits and try to get it from them. And if it happens to be in New York, yeah, you need more luck. Now the fact that we are a New York accredited lab, it makes life so much easier. So the sections are cut on our slides called the director slides. That's the only difference. The director slides are actually laser microdissection compatible slides. From there, we take in high-res image, digital pathology. It's all saved down the road. We might even scan that with an AI systems. Then the pathologist goes in there and just marks only the tumor area. That's it. We completely ignore all the vasculature, all the stroma, everything that we can do. Just marks the tumor area and then use the laser to microdissect the tumor area. Now, for our pharmaceutical partners that wants to play around in the tumor microenvironment, we can, of course, point the laser to the tumor microenvironment and look at the regions of interest for responders versus non-responders. But for the clinical workflow, we are specifically looking at the tumor areas. From there, shoot it into a tube, add our secret buffers, so to speak, and then from there, we move on and put it into a mass spec. So mass spec comes in two flavors, just discovery proteomics, where we are looking at 5,000, 6,000 proteins from a single section. That's more for a research side of research setting. For the clinical setting, we are looking at 72 known oncology biomarkers in one single go. So this is from two sections, two 10-micron thickness sections, and one H&E image. We're looking at 72 known quantitative biomarkers. From here, the data comes out. It's a medical director sign-off, and peer-to-peer, this report gets issued out in 5 business days. So the time the block gets in our house to the time it gets reported out is in 5 business days. Now, the clinical report, let's see if I have a picture of it. So the clinical report, this is really was created because a community oncologist said to us, I see 50 patients a day. If your report cannot speak to me in 15 seconds, forget it. So we redesigned the whole clinical report to red and green and estimate, hey, these are the agents that are likely to work, and these are agents that are less likely to work. What do we inform on? We inform on chemotherapy options, what we see. We inform on targeted therapy options, and also, well, immunotherapy options as well. So go from there. Why chemo? Now, this is a fact that a lot of people are brought out here today, they're stating how important chemo is. But to quantify how much chemotherapy is really given in our oncology community, there was a paper out, this was in 2020, I think, and it was looking at the data from 2018. That said 78.7% of folks are actually getting chemotherapy, they are chemotherapy eligible. Maybe let's say 4 years or 5 years down into it, now it's gone down to say, let's say 60%. So what does that mean? 3 out of the 5 people that walk into a clinic are going to get chemotherapy. Do we actually look at any of the chemo biomarkers when we are treating or giving recommendations? No, we don't. So this is, I guess, the elephant in the room. So why don't we measure the chemotherapy biomarkers? Give it to the oncologist. Let them make the decision. So the biomarkers assays that we look for, what do we look for? So I just broadly divided them into chemo and targeted clinical trial markers. Just going to spend some time on kinds of chemotherapy agents that we look for. Cisplatin, platinum-based regimens. In the previous slide, you've seen like 88. There are 88 cytotoxic regimens that is given in the world of cancer. But we don't really look for chemo biomarkers. So ERCC1 is a known resistance marker for platinum agents. Sure, there are other resistance agents, but we happen to put ERCC1 on it. Tubulin beta-3, again, a known resistant marker to taxanes. Now, of course, paclitaxel, nab-paclitaxel, and all that stuff. But this becomes important also in the space of antibody-drug conjugates. Antibody-drug conjugates, of course, it's binding to the cell, getting internalized. the killing actually happens because of the chemotherapy agent. Do we measure that target? Perhaps not. So tubulin beta-3 is one of the agents that we look for. We actually reanalyzed one of the clinical trials, the Ithaca trial, and looked at tubulin beta-3, and we could see spectacular difference in the overall survival of folks who were given a docetaxel regimen and not given a docetaxel regimen. That's just more like 1,000 days difference. Gemcitabine, I mean, many of the rest of the markers are just practically sensitivity markers. So gemcitabine gets into the cell through HENT1. So it's a transporter to get into the cell, and then it gets negated at some point down the road with RRM1. Topo1, now it's much gained in popularity because of the approval of several ADCs like trastuzumab deruxtecan. But Topo1 has been used, I mean, our irinotecan and topotecan has been used forever in glioblastoma. It kind of got deprecated out now. But we have seen 15% of glioblastoma patients actually have high levels of Topo1. And we have case reports of where physicians have treated them with irinotecan refractory glioblastoma responding to this. Topo2A for all of the rubicins and the etopicides, FR alpha. Now it's got approved because of a newer agent that is approved, but you could potentially use methotrexate or pemetrexate. 10% of methotrexate does go in through FR alpha. MGMT, a marker for resistance to temozolomide. I think in the colorectal cancer space, what's happening lately is that there are some trials that are coming out, like the Maya trial, where they're using temozolomide, which is a brain cancer drug in colorectal cancer, to induce mutations, increase the tumor mutational burden, and to go after it with anti-PD or PD-1 drugs. The data is looking good. Then we heard this morning about the TYMS, thymidylate synthase, but along those pathways from the 5FU pathway, before it goes and inhibits the TYMS, there is the thymidylate phosphorylase. We have seen data, and we published on this, where we can look at thymidylate phosphorylase levels and kind of predict who will respond to a 5FU therapy or not. Now, on the targeted agent side, I mean, there's the classical translocation markers, ALK, RET, and TREC. But more importantly, we work with the folks with MSK, looking at HER2 and low HER2 populations. We are also looking at TROP2 across so many cancer indications. Then since our work is out in the community, and there's a lot of community oncologists sending us, and community oncologists is also a very good recruiting place. One of the community oncologists in Arkansas is using our test to figure out what the levels of AXL is in his sarcoma patient, so that he can put the patients into an AXL clinical trials. I can go on and on, but in the interest of time, I'm going to move on to the next one. So I can show you all the papers, but one thing that we recently published in the pancreatic cancer space was to take a look at what we have seen in the CAP-CLIA environment across so many cancers. And we see that this population of HER2 that is overexpressed in pancreatic cancer. In fact, we have a paper coming out of where the patient was treated with trastuzumab deruxtecan, had cancer vaccines, a whole host of things, and this patient's alive 4+ years out now. We're quantitating the conventional chemotherapy targets, but also the newer agents, newer antibody drug conjugates, one being a folate receptor alpha. That's historically not thought of in pancreatic cancer space, but with 10% of pancreatic cancer does have folate receptor alpha. In fact, one of these individuals in here has extremely high levels of folate receptor alpha that you could potentially treat or influence with methotrexate. Encourage you to look, well, the product line, by the way, is called OncoOmics DX, so OncoOmics DX, and it's a whole list of publications. These are all work that we have done with folks with MSK, with University of Chicago, Hopkins, and all other places. With that, let me stop.
Great, thank you.
Thank you.
Very much. Thank you.
So I'd just like to thank Steve and the organizers for bringing us all together today. It's been a great meeting so far, and I've personally learned a lot. So just by way of introduction, I'm an associate professor in biology at MIT, where I work closely together with colleagues like Mike Yaffe, and I'm also a gastrointestinal pathologist at MGH, where I work closely with my oncology and surgery colleagues such as James Cusack, Dave Ryan, and Keith Flaherty. So when I started my lab in 2014, we really focused on two topics. One was trying to understand the effects of environment and diet on colon cancer initiation and progression. And the other part of my lab really focused on developing more physiologic models to study colon cancer progression. So as a pathologist and as a researcher who's interested in better modeling colon cancer, what factors should we consider when modeling human cancer? So when we get specimens in pathology, we get pieces of colon or appendices that have these masses, that have these tumors shown here, where in the colon, for example, we have these fungating lesions that emanate from the wall of the colon and invade deeply into the colon, where these cells can eventually metastasize to distant organs, lymph nodes, and in some cases break through the wall of the colon and intestine and seed the abdominal cavity or peritoneum. Another important factor when developing models for colon cancer or appendiceal cancer is to make sure that the tumor cells that we are modeling, that the tumor cells in our model reflect the histology or what the tumor cells look like in the patient. And most colon cancers and a subset of appendiceal cancers are adenocarcinomas, meaning that they're these glands of tumor cells that invade into the wall of the colon. And lastly, what's been more challenging to model in the lab are ways in which colon cancer, appendiceal cancer, and other types of cancer spread throughout the body. And there's really, for these steps of colon cancer progression, there's really no surrogate to mice, where, for example, colon cancer arises in the, So colon cancer arises in the colon, can enter into the vasculature and seed distant organs such as the liver and lung. The appendix here, a subset can invade into the vasculature and spread to distant organs, but most appendiceal cancers and some colon cancers can break through the wall of the appendix and colon and seed the peritoneal or abdominal cavity. And really to study these mechanisms, again, we've been interested in using more robust mouse models. So one way in my lab, and as others have talked about today, is to use organoids, where we can take, for example, biopsies from mice and patients from their colon and grow normal organoids. So these are three-dimensional structures that reflect the histology of the organ from which they're derived, and they're grown in a matrix called matrigel. And what one can do in the laboratory setting is take these organoids and then turn off or on genes that are associated with the cancer that one is interested in studying. So for example, in colon cancer, we turn off a gene called APC, and we activate a gene such as KRAS that drives a lot of the biology behind colon cancer. An alternate route for generating these tumor cells is to take biopsies from patients and then grow tumor organoids directly from the patient's samples. And what we're interested in doing is, in my lab, is we've really leveraged mouse colonoscopy approaches, where we can do colonoscopy in a mice and we can transplant these organoids, whether they've been derived from normal tissue or from cancer tissues, implant them into the wall of the colon. And over a period of time, these transplanted cancer organoids give rise to a primary tumor, and a subset of these mice develop metastatic disease, such as to the liver, and in other cases, these tumor cells again can break through the wall of the colon and seed the peritoneal cavity. So here's what these primary tumors look like in the mouse colon, and here's just a representative example of what these lesions look like in the liver. So this was really an important development for my lab and for others, because it permitted the rapid modeling of tumor initiation, progression, and metastasis. So in my lab over the past few years now, we've been trying to leverage this approach to discover new therapeutic targets. So we performed a very simple experiment. I only have 2 slides here to highlight work that we've been doing in the last 3 years, but I think it's really interesting in the sense that we took samples from patients, so colon biopsies, or we took samples from mice, normal mouse colon. We using genetically engineered approaches, we turned on genes that are commonly found in colon cancer to generate these, organoids that are cancer in a dish, but they've never been a cancer in the in vivo or mouse colon environment. We then transplanted these, what we call naive colon cancer organoids, into the colon of a mouse, and after about a month, these transplanted organoids formed primary tumors from which, again, we generated organoids. So although these organoids are genetically identical, these naive organoids had to undergo some type of adaptation to adapt to the colon environment, which isn't present in our culture conditions. And this was really powerful because this enabled us to identify an important gene in colon cancer that we think plays an important role in regulating how these colon cancer cells interact with the immune environment. And this is really important because over the past decade, we've known that immune checkpoint treatments such as Keytruda for Merck and others have really been game-changing for a subset of cancers, such as for melanoma and a subset of lung cancer. But the vast majority of colon cancer is resistant to these checkpoint inhibitors, and we think that SOX17 really plays a critical role in how these colon cancer cells interact with their immune microenvironment. And just one more slide here. In biology, one way we test for the function of a gene is to either turn it on or off. So we performed a simple experiment. We took our colon cancer organoids, we turned off the gene SOX17, and then retransplanted them back into the colon of recipient mice. And what you find here is that in contrast to the control colon organoids that have intact SOX17, 80% of the transplanted mice form tumors. When we now turn off SOX17, less than 10% of the recipient mice actually form tumors. And what we think is happening here mechanistically is that SOX17, both in mouse and human colon cancer cells, is turning off a pathway commonly used by both normal cells and cancer cells to interact with the immune system. So we think that SOX17 plays an important role in regulating how these colon cancer cells regulate tumor progression. And we think so it does this by regulating how these colon cancer cells interact with their immune microenvironment, particularly this particular subset of immune cells called T cells, which are known to eliminate cancer cells. And we're interested in testing, for example, SOX17 and other tumors of the GI tract, and I think the appendix would be a great model to test this, where at least in some initial studies, we believe that a subset of appendiceal cancers do express high amounts of SOX17. So I just want to summarize and say that we've been very interested in leveraging this colonoscopy-based approach to study both mouse and human colon cancer, and we think that the power here is that this type of approach allows us to rapidly model predicted cancer-associated genes. Working together with Mike Yaffe, we're interested in testing his approaches for studying combinations of chemotherapy in patient samples in the mouse colon. We have models where we can test for neoadjuvant radiation in the colon. As I demonstrated to you the example of SOX17, this is really important tool to study how the tumor interacts not only with the immune cells, but with other populations such as the stroma and vasculature. We can also do these types of transplantations in mice that have a humanized immune system. And lastly, something that my lab has been interested in, which we haven't talked about today, is these types of approaches now allow us to understand how different physiologies and the environment, such as obesity, impact tumor initiation and progression. Thanks.
Thank you, Omer, and thank you to all of our speakers in this second section. I'm opening up the floor for questions.
For Dr. Flaherty, certainly uveal.
Could you use the mic?
Okay. Certainly uveal melanoma is rare, falls within the rare. So is there a sequence of studies or progress that you can sort of delineate that led to the successes that you have?
Yeah, we actually did finally get a therapeutic success in uveal melanoma that I skipped over, which is this GP100 CD3 bispecific. But we still, continue to cajole companies and try to figure out a strategy for, sharing risk and cost for GNAQ/11 development, at least in clinical development, which I think we might be able to do through this foundation that I was alluding to. I think, as I mentioned, first and foremost, people have to come together. So that started in melanoma. I mean, before the therapeutic successes of cutaneous melanoma, just before, and coming out of them in the early 2010s, we managed to be able to kind of corral not just U.S., but largely U.S. centers, basically suggesting that we needed to put our shoulder together in terms of trying to figure out mechanisms of action and resistance, really, to map kinase pathway targeted therapies, which for BRAF mutant melanoma, and for PD-1. And now, if you look at the published literature, you'll see the outcome of that, which is that basically we've been sharing samples, sharing data across a network of now 20 centers in a way that's really been able to accelerate our ability to understand, kind of some of those issues that, again, cutaneous. So under the umbrella of the same foundation, is where we launched the Aguirre uveal melanoma, foundation. Basically, it's got its own name with a kind of overlapping scientific community, but the same clinical investigator community. And so we've been doing the same work now across that network of sites in terms of trying to understand the immune microenvironment in liver, because uveal melanoma metastasizes the liver and nearly always causes its fatality there. So that's kind of a particular focus and problem. Non-immune microenvironment also in the liver, which I'm trying to understand its influences. And so I would say we hope we've been doing the work that actually paves the way for therapies that we think would be uniquely useful there, while we keep holding our breath on the GNAQ/11 drugs as at least a foot in the door, knowing that cancer's complicated, you need combination regimens. So anyway, that just completes what I was opening with there in terms of the benefits of trying to corral or define first a community and then corral folks in it.
So the community was first. You got all the
Absolutely.
melanoma experts, and they
Absolutely.
said, "Here's 1% of our disease."
That's right.
Yeah. Thanks.
Yeah.
Do you mind introducing
Jim.
Oh, I'm Gary Kelloff.
Thank you. I'm Jim Palma. I'm the Executive Director of Target Cancer Foundation, and we're a rare cancer research and patient support foundation just across the street in Kendall Square. And Dr. Flaherty, I was really interested to hear your remarks about NCI match and the high number of rare tumor patients that were enrolled in that study, but certainly not surprising given the lack of clinical trials available for patients with rare cancers. Certainly, it's sort of an idea and a problem that we're pursuing at our foundation through a trial that we're running called TRACK, which is enrolling patients with any rare cancer, getting them NGS, liquid and blood NGS at no cost, and then combination treatment recommendations from a virtual molecular tumor board. So for us, we've seen a lot of promise in that, and we've seen 10 patients in our study with appendiceal cancer, which may have been difficult to otherwise accrue. But it seems like the research infrastructure and certainly the clinical infrastructure is still very much histology dependent. So I'm wondering your thoughts as you look ahead post-match, how the spirit of that study and biomarker-driven treatments, and especially the impact that they have in rare cancers like appendiceal, can be brought forward and continued.
Yeah. And in my comments, maybe I supported this idea of site of origin as the altar at which we worship. And just to be clear, I spent my entire career trying to erode that and focus on biomarker-guided drug development, focusing first on somatic genetic alterations, but now moving forward, even with immunotherapy response predictors, trying to break down, those barriers. So some rare tumors will benefit more or less from that concept, right? So I was trying to suggest that like KRAS is kind of the example for appendiceal carcinoma, that KRAS is not going to cure hardly anybody. KRAS targeted drugs, right? But to be totally frank, but I've always made this case that you need a foot in the door. And so for the significant fraction of patients with appendiceal who have KRAS mutations relevant to the specific drugs that are coming along, that's the cross-cutting opportunity, like where those drugs are going to be developed because of the prevalence in lung cancer and colorectal cancer, plus others. And so draft off of that and try to get some inroad there and understand the potential organ site-specific issues in terms of why appendiceal carcinoma might be more or less resistant. I mean, we already know that colorectal cancer is somewhat resistant to G12C KRAS targeted drugs relative to lung cancer, and probably borrows from some of the BRAF resistance biology that I and my group have been working on for years now, but it still allows us to actually assemble, in that case, BRAF, EGFR combination therapies based on that understanding, and actually, get regimens to market and for patients. So I mean, I'm just focusing on your question in terms of what's the low hanging fruit or the path of least resistance. It's try to nest appendiceal cancer subsets, with their other molecularly defined subsets that are represented in other cancer types. So KRAS, GNAS, at least from my scanning of the available whole exome sequencing data, the others are kind of pretty small, very small subpopulations. But it's not all about DNA sequencing. I mean, this is where, nor is it all about RNA sequencing, nor is it all about proteomics. And functional diagnostics. You have to throw the kitchen sink at the concept of trying to direct patients to initially investigational therapy and find out who responds.
Great. Yes, go for it.
Well, Maggie Senthil, I'm a surgical oncologist at the University of California, Irvine. I've enjoyed both the sessions, and the comments that I'm going to make applies to both of the sessions that we have heard thus far. And I really think, many of you have highlighted this, the three major challenges that we have in managing disease, not just in appendix cancer, but peritoneal carcinomatosis in general, is lack of the diagnostic, predictive, and prognostic biomarkers that are readily and easily available, lack of appropriate imaging studies that are able to assess response to treatment, and finally, lack of innovative therapies that are able to modulate the tumor microenvironment in the peritoneal space. Now, over the course of the several decades, we have tried to perfect the surgical approaches to the management of peritoneal carcinomatosis, and a lot of the peritoneal treatments have largely focused on treatment after cytoreduction, so mostly as an adjuvant approach. But it seems like there is a great opportunity to be able to take advantage of iterative treatments prior to considering surgical approaches, where we have the best chance of modulating the tumor microenvironment in the peritoneum, and try combinatorial therapies, whether it is bidirectional, unidirectional, and modulation of the peritoneal microenvironment before we even embark on surgical treatment approaches, and possibly even convert patients who are not surgical candidates up front to cytoreduction candidates, which might improve overall their survival and possibly, for many diseases, not just even in appendix cancer. Now, over the course of these several talks, we have heard that many, or at least few of the institutions, are looking at normothermic IP paclitaxel with combination of several other agents, and they're being done in single institutions. And I believe the reason we are here together is to figure out as to how we can accelerate these kind of novel approaches by including multiple institutions to participate in studies like that, and using these in vitro tumor models to also inform about which drugs would be best used. So my question to the group and the panel is, what would be the next appropriate step for us to think about the most important studies that are being presented here to take it to the next level and have multiple institutions participate? And where would the funding come from for that kind of multi-institutional studies? And this could be an accelerated phase 2 studies that could immediately impact patient outcomes. So that would be my question, both for the audience as well as the panel.
Well, I'm going to take a quick stab at the financial modeling. We have an entire session this afternoon that's really going to focus on, I'm going to use the D word again, a disruptive financial model that allows this true collaboration, especially across the academic groups. But to the panel, any comments?
I would say one of the things you would like to have is an RFA from NIH. And where you get an RFA is you put together a small conference of people who are interested in the field, the stakeholders, and then you write a report on that. And then NIH can sit on that, decide whether they feel that this is worthy of an RFA. But that would be the mechanism, I think.
But isn't this the RFA? Aren't we here doing that right now?
Well, yes, but NIH needs to say, okay, we're putting aside a pot of money. We would like to have applications focused on appendiceal cancer or this group of rare cancers, and we would encourage groups to come together. So that is the way generally you nucleate people around something, is put a pot of money there,
right,
and groups will then write applications.
You need a source of financing Which may
So-
not be the RFA, but it
Exactly.
may be mentioned.
So, I mean, in this room, in this
room.
room, we have NCI, we have our collaborative researchers, and we also have some pots of money in this room. And I'm looking at some of my VC partners here as well. I think this is the RFA. This is the call to action. I'm going to take one more question, and then I'm going to, Mike. Oh, hang on. I've got JP. Do you want to duke it out?
Let me just, mine will be very quick. I just want to build on something that Keith said that's really important. It's easy to get samples, not easy, but you can get samples for genomics. But what I want to point out is everything that Keith has said about RAS and EGFR, those are signaling molecules. And if we don't get phosphoproteomic data, then we're not necessarily going to be getting at the answer. And there's something really unique that we can do in the setting of peritoneal carcinomatosis that is much more difficult to do with primary solid tumors. With primary solid tumors, the pathologist owns it, and the phosphoproteome degrades relatively quickly. With peritoneal carcinomatosis, we can snap freeze, or you can snap freeze some of these samples right away. And that now will give us data that will be very insightful. So in building on what Keith is saying, let's not just as a community, let's not stop at genomic and transcriptomic data. Let's make a real push for proteomics, but especially phosphoproteomics.
Great. JP, do you want that last
yes,
to last word? Because I'm going to have the last word. No, go for it. You sure?
The question I had is, very the mouse, the VMT model, looks remarkable. One question though, does it kind of model the kind of depot effect that we see in the peritoneum, and also kind of the blood peritoneal border? Like one interesting study that Michael Foote showed is that P53 mutant tumors tend to have more infiltrative invasion versus P53 wild type, and like at that level, would you see that sort of interaction between tumor and membrane in your models?
That's a great question. It's the exact reason we've developed it. It's brand new. I mean, we haven't published it. What you're seeing there are hot off the press pictures. So those are the kind of questions we're going to ask. Right now, we've done it with Colo 205s, who are going to start. We're starting to collect patient tumors now. And so that's what we'll look for, is whether it's just going to stay on the surface, whether it invades, whether it does both, and then which of those cell populations is more susceptible to intraperitoneal therapy versus vascular peripheral therapy. So great question. It's exactly what we want to look at.
Great, thank you. I'm going to do try and briefly summarize, close out this morning. What have we heard about? We've heard about our surgical approaches. We've heard about the importance of germline characterization. We've been talking a lot about organoid development, and also Omer's work on organoid transplant going into animal models, understanding how that affects the primary and the metastatic tumor environment. We've been talking about de-risking trials using MPS, personalized tumors on a chip, targeted proteomics to really understand the tumor in the patient and also the microenvironment. We have talked about tumor profiling with very specific therapies. The technology use of mRNA delivery, especially for delivering immunotherapy, targeted radiotherapy. Thinking about single patient INDs, moving away from these big clinical trials and really focusing on an individual patient. And the word that I've heard a lot about this morning is community. And I think, we have a community here. We are this community. We need to bring in our patients, and we need to bring in our caregivers as well to make sure that they're part of our community. Before I go to a video that I want to share with you, I want to mention a number that Kevin mentioned. 40% of cancer is rare. That's what we've heard this morning. And I think that's a very powerful message for what we're thinking about here. So with that, I'm going to introduce you to Priscilla Thompson.
So my name is Priscilla Thompson, and I became familiar with appendix cancer through my sister, Kathy Maffei. She was diagnosed in 2018. We thought she was going in for a routine appendicitis. She'd been hiking up in Canada, and we found out it was appendiceal cancer, which I knew nothing about. and we knew nothing about. So going down the gamut hole of what appendiceal cancer was kind of confusing for all of us. We all dug into research. We were reviewing a lot of things, trying to figure out what was the next treatment. And we went to an oncologist, and the standard treatment they were offering was just a cookbook recipe of how you treat colorectal cancers, which, as we found out retrospectively, appendix cancer is not just simply colorectal cancer. It is its own beast. It has many layers. So we decided to go up to UPMC to a physician up there who was known for high-tech surgeries, because that's where our research took us. And my sister had high-tech surgery up in UPMC with Dr. Bartlett, and she did well afterwards. It was a vicious surgery. She did recover well from it. But the following up on that was just, again, cookbook chemotherapy with Aguirre or Flaherty chemotherapy, which is just what they do with regular colorectal cancers. So again, we dug deep into the research with colorectal cancers, and we stumbled on something from Dr. Aguirre, who is a part of a big consortium in the world with Beacon trials, which targeted specific mutations. I also came upon ACPMC, which is the Appendix Cancer Society, which helps guide you with off-label type of treatments for colorectal cancer. And I became friends and met with online one of the people who work with very deeply with that, Deborah Sheldon. And she kind of gave us pointers on, I didn't know that you needed to have, a whole genetic workup when you had your cancer surgery done, that the pathology meant a big difference in how you treated these cancers. No one told us this. None of the oncologists we saw multiple oncologists we saw them at Penn State, we saw them up in Sloan Kettering, we saw them at UPMC, and no one recommended anything else but that. But finding other people who were connecting through this horrible disease helped guide us with my sister's treatment. And so she did land up going on targeted therapy, which was specific for her genetic mutation. So I think the biggest takeaway that we found is that this should not be a cookbook. And sometimes you have to think outside of the guidelines. We were offered clinical trials at MD Anderson, but with the clinical trials, we didn't know what we were getting. And, they gave her 6 months in 2018. They said you had 6 months to live. My sister passed away January 7th, 2023 of this year. And it was a long battle to get where she was, but she did have some good quality of life in those times. And so what I would say is with appendiceal cancer, you can't treat it like the run-of-the-mill colon cancer. It's not. It's different. There's many layers to it, and some people have different types of appendix cancer, whether it's signet cell or adenomucous carcinoma. Many people have different types of ways to treat it, but the standard treatment doesn't work, and there needs to be more research and more clinicians willing to take the gamble of saying, this is what we have for you. You're not going to survive it, but we're going to just throw it at you anyway, and show them some of the clinical trials that are out there and let them make the decision, make the patient be their own advocate, make the family members be their own advocate, because without that advocacy, my sister would not have lived until 2023. She wouldn't have seen her grandchildren born. She wouldn't have gotten to know them. She wouldn't have been able to go on hikes up in New England with myself and my husband. She wouldn't have been able to see a lot of things in her life that she got to see while dealing with this devastating cancer. So that would be my takeaway is, don't just go cookbook on this. This is not a cookbook type of cancer.
We are going to break for lunch. Lunch is being served outside. I would like you to bring it back in here. Stay where you're seated. And thank you very much. Thank you for everybody who's participated in this morning's session. Thank you all for being so interactive, and enjoy the break.